Hi there, my name is Brodie Badcock Parks, and I'm an incoming master's candidate in the Department of Psychology and Neuroscience at Dalhousie University, and I'm really thrilled to be with you here today to chat a little bit about the topic of my undergraduate honors thesis, where I looked at seasonal differences in the brains of European starlings. Now, when I talk about seasonal differences, or seasonal variation, we're often used to thinking about spring, summer, fall, and winter. But in birds, at least for the purposes of this presentation, I've distilled it down to a binary. Birds are either in breeding condition, which for birds here in North America typically lines up to spring and summer, when there's a longer photoperiod, or they're in non-breeding condition, which typically lines up to fall and winter, when there's a shorter photoperiod. And just by looking at these photos of European starlings, you can see that there are several clear physiological differences between breeding and non-breeding birds of this species. For example, the starling on the left in breeding condition has much more iridescent plumage and a different colored beak. And even though we can't see them in this photo, we would also expect this bird to have larger, more developed gonads. Now, of course, there are also behavioral differences between these two birds as well. First, we would expect the starling on the left, in breeding condition, to produce longer, more complex songs during breeding season in order to attract mates. And starlings in non-breeding condition, much like the bird on the right, tend to be much more gregarious and social. And this is why, especially during the late summer and fall here in North America, we see large clouds or murmurations of starlings, especially in the evening. Now, these changes in physiology and behavior in birds are also accompanied by changes in the brain. And in this study, we focused on two major brain regions. First, the hippocampus, which is broadly involved in a variety of different spatial behaviors, including spatial learning and memory. And seasonal changes in hippocampal neurogenesis, that is, the production of new neurons in the hippocampus, have also been associated with the onset of migration, as well as increased exercise. The other major brain region we looked at is called HVC, and this is the largest nucleus of what we call the song control system, which is broadly involved in listening to, learning, and producing birdsong. HVC is really involved in that production aspect of birdsong, and the majority of studies to date have shown a significant male bias in both the size and the amount of neurons in this area, given that typically male songbirds are the ones who produce much more birdsong during breeding season in order to attract a mate. Now, because day length is the main cue to determine breeding or non-breeding condition, birds can be brought in and out of breeding condition quite simply, just by changing the amount of time the lights are on in the lab. And most studies studying seasonal differences in birds use this method. The problem here is that that's not really at all natural. The birds are in captivity, and all of the factors associated with being in the natural environment are eliminated. So to solve this problem, in our project, we simply captured birds at different times of the year, at different levels of natural photostimulation, and we sacrificed them quickly, without ever having to introduce them to the constraints associated with the lab. Another issue is that a very common marker of neurogenesis, bromodeoxyuridine, or BRDU, is an exogenous marker, meaning that injections of this chemical are required in order to tag new neurons. And this is relatively invasive, it causes stress to the bird, and it potentially suppresses neurogenesis. So in our study, instead of using injections, we used immunolabeling to tag new neurons for an endogenous marker of neurogenesis, doublecortin, and this is a protein that is only expressed in new neurons for about up to a month post-mitosis. Here's what doublecortin staining looks like in our two regions of interest, on the left in the hippocampus and on the right in HVC. After staining, we hand-counted the number of doublecortin positive cells per field of view in both of our regions, and then we compared our counts between breeding and non-breeding starlings. In the hippocampus, we did not find any evidence of seasonal differences in the amount of new neurons in this area. And this might be because, to date, studies looking at seasonal differences in the hippocampus have been done primarily on what we call hippocampal specialists. That is, species that exhibit pronounced hippocampus-dependent or spatial-dependent behavior. For example, food-storing birds like chickadees, brood parasites like cowbirds, and long-distance migrants like sandpipers. Now, starlings, on the other hand, they're not food-storing birds, nor are they brood parasites, and here in Nova Scotia, where we captured them, they're generally considered non-migratory. In our other area of interest, HVC, we did find an influence of season, as well as a sex difference. We found that breeding female starlings had significantly more new neurons in area HVC compared to males. And this goes against much of the previous research to date, which has shown a pronounced male bias in this area. However, one recent study using wild-captured and non-lab manipulated icterids also showed a similar female bias. When we then compared the number of new neurons in HVC between both brain hemispheres, we found a significant left hemisphere bias in breeding birds, suggesting that during breeding season, the left HVC nucleus preferentially recruits new neurons. And while prior studies have shown evidence of HVC lateralization, including lateralization of neuronal activity, this would be the first evidence of lateralization of new neurons using Doublecortin immunoreactivity in songbirds. In short, we took a more ecologically valid approach to studying neurogenesis in starlings, and we ended up getting different results than most of the previous studies to date. We found no differences in the hippocampus, a female instead of a male bias in HVC in breeding starlings, as well as evidence of left lateralization in HVC. And given that more and more research is showing that even brief periods of lab captivity can alter the songbird brain, our results emphasize the importance of using a more ecologically valid field-based approach when studying the brain and behavior of songbirds. Wrapping up, big thanks to all of my lab for helping me with this work. And if you have any questions for me, you can join me July 31st for a live Q&A, or you can reach out via social media or via email. Thanks very much for your attention.